for introducing your very ambitious project. And I think we can continue in writing down your questions into the chat because we have to move on. So our next speaker is Mark Blaxter from the Sanger Institute. Mark?
Yeah, I'm here and I will share now. This one. So
yes, we can see it.
Yes, slide. Okay. Okay. So thank you, Neil, for introducing the Tree of Life project, and thank you all for hanging around to listen. And thank you for the invitation to talk. I'm probably not going to talk about any protists at all, although I am a card-carrying leishmaniac, as I did my PhD in leishmania and have carried a torch for leishmania and other interesting trypanosomatids ever since. So I'm based at the Tree of Life, and we have at the Tree of Life a science program called, I'm based at the Sanger Institute. At the Sanger Institute, I have a program called the Tree of Life. Tree of Life has the ambitious goals of generating high-quality reference genomes for as many species as is possible across the Tree of Life, and we do mean across the Tree of Life, so including all the phyla which are classed as protists. And to do that, we're going to have to do some new tech. We're going to have to get some in the wet lab. We're going to have to get some new tech in the bioinformatics lab. And then we're also going to have to share these data and do some science with it. And so just like Neil, I'm impressed by mega projects like the Hubble telescope and like the Hadron Collider. But I think the biggest mega project ever has been the Linnaean project. This has generated 1.9 million species identified thus far. And the exciting thing for me in this is that these data are openly accessible to everyone in the world. They're a lingua franca in which we can talk to anybody, anywhere about species. Because of that, we want to change this from being a catalogue of life, where we have the names of all the books, through to being a library where you can actually take the book out and read the books of life. Okay. So of course, most of life, as Neil pointed out, is small. This is from the Wellcome image collection, stuff that was seen in the water in the River Thames in London under a microscope. And of course, again, showing the tree that Neil showed, this is because most of life is actually in these microscopic organisms. So this is a real challenge to our project. At the Tree of Life, we have some major projects running. One of them is the Darwin Tree of Life project. That's what I'm going to talk about mostly. But another one that must be of interest to people here is our aquatic symbiosis genomics project. This is funded by the Moore Foundation to sequence about 1,000 symbiotic systems. And these are going to include lots of protist and single-celled systems where protists have made neosymbioses with photosynthetic or other partners in order to access new biochemistry and new niches. And so expect some interesting data on protists to come out of that. That project is just starting. We're just onboarding partners. The announcement for, well, the request for applications is just closed. We're going to be picking them. So that's going to include mega biota, animals and plants, as well as many, many of the protist-sized microbiota. The major project, which has now been running for a year, is the Darwin Tree of Life project. As Neil mentioned, this is a collaborative project amongst institutions across Britain. And our idea here is to generate a reference genome for every eukaryotic species that's been described in Britain and Ireland. So this is a huge task. It's 60,000 or so species. We're going to start with this idea of a phylogenomic wave that Harris introduced earlier. We're going to start with the families. And interestingly, the families in Britain is about 4,000 different families of eukaryotes. That corresponds to about 40% of all the families of eukaryotes on Earth, just the way that phylogeny and ecosystems overlap. So we'll be able to contribute about 40% of that first phylogenomic wave to the Earth Biogenome Project. Inside that set of 4,000 families and 60,000 species are not very many things that we'd call protists because they've been poorly described taxonomically. And I counted 123 species of Euglena zoa on this current checklist. And as Neil points out, there are going to be many more. So I'm pretty confident that we'll be able to generate good genomes for these, even if they are entertainingly triploid or aneuploid or interestingly ploidy. And I was going to go through how we're doing that quickly. So there's two kinds of data we're going to generate, genomic data we're going to generate. One is what I call long read data. These are primary sequence data from individual molecules where the actual sequence length is long. So we're aiming for average lengths of well over 15 kilobases. So if you think about the current Euglena assembly, even with Neil's team working really hard at it, the assembly is at 2 and a bit kilobases after a lot of assembly work. We're aiming to generate data which have much, much greater primary lengths than that. The other sorts of data we're generating are long-range data. These are data such as the 10x data that Neil introduced, which give us links between reads or sequences, which allow us to link molecules together to scaffold. So I'll just quickly show some of the long-read data. This is from the data we generated in the last year. So this is 253 different species. There are a couple of single-cell species in here, not Euglenads. Showing that we're getting an average read length of about 13 or 14 kilobases. But the important thing is these reads are as good as the best quality Sanger reads or the best quality Illumina reads. So they are extraordinarily high quality. And this allows us to generate very high quality genomes. The other sort of data, this long range data, is probably less familiar to people. What we know is that in a nucleus, and this happens to be a human fibroblast, but imagine it's a Euglena, in the nucleus of a cell, the chromosomes aren't smeared out all over the place, but they actually sit in relatively tightly compact domains. And therefore, if we have a mechanism, which we do, a biotechnological mechanism of cross-linking DNA such that only DNA which is close together in the nucleus will be ligated and sequenced together, we can generate a map of the closeness of DNA in 3D space, and that corresponds to the closeness of DNA on the chromosomes, and so we end up with chromosomes. So this is called high-C, or conformation capture sequencing. This is in humans. It can be done in any species. We've done it in a large number of animals and plants now. And what it does is it turns the data which we get out of the long reads, which is on the left of this slide, which are very good, very long contigs, into something which maps together into what are mostly chromosomes and some pieces. So in these pictures, the density of red color tells you how many links there are between sequences. And to go from the left to the right, we've used the high-C data to scaffold the sequences. So this allows us to go to very high quality genomes very quickly. The other thing we want to do in the Tree of Life project is not ignore what else is in the box. So when we sequence something, there are many other things in the box of that organism that we're trying to sequence. And so this includes mitochondria and chloroplasts, but also other things, symbionts, parasites, and other hangers-on, which we call cobionts. So we're trying to separate out these as well, so we get all of the genomes that are in the box as separate assemblies. The other thing to say is, wherever possible, we're trying to sequence from individual organisms. So for protists, this is difficult. No doubt about it. A single organism is a single cell. For metazoa, we can choose an organism and we can get several thousand cells. But the sort of stuff that Neil is doing, trying to do microcultures, etc., and sequencing from clonal isolates, should allow us to do all these technologies on protists. And this is an example. You'll notice that this isn't a protist. This is a moth. It's not a particularly big moth. But from this individual moth, we've managed to generate not only a good estimate of the nuclear genome, which is now scaffolded into chromosomes, but also its mitochondrial genome and the genomes of 3 symbionts, 3 bacteria that were living inside this moth. They're called Wolbachia. They're quite common in insects. 3 different Wolbachia genomes, each about 1 million bases long, also from this assembly. So that's really important. We're going for the whole organism and doing it from individual cultures or individual isolates. The other thing we're doing, as Harris said, is to make this data open. And we've all signed, as Neil showed you, we're going to release all these data openly to the world. We've started that. So there's a QC site where you can see all the QC data. So you can see all the sorts of data we have, all the sorts of assemblies we're producing. And I've put these 2 slides the wrong way around, have I? No, that's all right, I did. Okay. So, and we're also releasing our interim assemblies. So these assemblies that we've got, as on the left here, before we've done this scaffolding and all the curating, so that you've got them before they're released to ENA. The other thing we're doing is trying to publish these data as quickly as possible. So there's no point in saying we've got a beautiful assembly of species X if that is only on our hard disks. And there's no use in releasing it to the public databases if nobody can cite it, if nobody's sure if they're allowed to use it. So
Mark?
I'm going to stop this last slide.
Okay, thanks.
I'm watching the time. Is we're also publishing these openly. This, again, is not a protist. This is a squirrel. But this is an openly published genome note, which just publishes the genome straight away so that you can use it. So our project is aiming to deliver these data for public use as quickly as possible. So these are our projects. This is what we're doing. And I've shown you the sorts of technology we're going to use to deliver it.